Cells are the foundation of life and the building blocks of our body. Every highly specialized cell in our body, such as brain cells or beating heart cells, begins as a tiny solitary blank canvas full of potential and possibilities. Cells that change and mature to take on their own distinct form and function. Many diseases are caused by malfunctioning or damaged cells. What if, instead of just treating disease symptoms, doctors could restore function and health by replacing cells that were lost or damaged by a disease? This is the basic idea of cell therapy. It transfers cultivated and engineered cells to patients to repair or replace degenerated cells, or repurpose them to enhance their function. One of the focus areas of Bayer's cell and gene therapy strategy is induced pluripotent stem cell-derived, iPSCs for short, cell therapy. This was brought into the company with the acquisition of Blue Oak Therapeutics. To better understand the technology, let me first introduce you to the terminology. Stem cells are undifferentiated cells that can develop into specialized cells. These specialized cells are used to form tissues and organs and to maintain their function, such as brain cells or heart muscle cells. In principle, there are two different types of stem cells: embryonic stem cells, which are isolated from early pre-implantation embryos, and adult stem cells, which are found in various tissues. While both types are stem cells, they have different potential. Embryonic stem cells are pluripotent, which means they can theoretically form any cell type of the human body. Adult stem cells, however, are multipotent, which means they can only form cells of a certain tissue, for example blood or skin cells. Induced pluripotent stem cells are also pluripotent stem cells and can be directly generated from mature adult cells by reprogramming. They resemble embryonic stem cells and can therefore be differentiated into almost any cell type of the human body. So how do we create iPSCs? In the first step, adult cells like skin or blood cells are isolated from a patient or a donor. These cells are highly specialized and have already deactivated their stem cell programs by switching off specific regions of their genomes. By now introducing specific genes into these adult cells, we can make these blocked off genome sequences accessible again. As a consequence, the adult cells travel back in development and revert to a pluripotent stem cell state. This induction of pluripotency is called reprogramming. These induced pluripotent stem cells can now be directed to form specialized cells with distinct functions, such as heart muscle cells or brain cells. By exposing the iPSCs to specific culture conditions and growth factors, they leave their stem cell state and form specialized cell types. iPSCs hold great promise in the field of regenerative medicine. Besides bypassing the need for embryonic stem cells, they can propagate indefinitely and give rise to almost every other cell type in the body, thereby representing an unlimited source of cells that we can use to replace those that were lost or damaged in diseases. So now, if we use these cells for therapy, we have two options. If the cells for the therapy are derived from a specific patient, this is called autologous cell therapy. In contrast, we are working on allogenic, or so-called off-the-shelf therapy, which is based on donor-derived cells. With this approach, one allogenic cell line can be used for all patients. This has great advantages regarding manufacturing complexity, costs, and comparability between different patients. Our iPSC platform is built on the technology of Blue Oak Therapeutics. Blue Oak has a superior understanding and know-how of the intricate requirements needed to differentiate the pluripotent cells into specific cells. By using sophisticated manufacturing techniques, we create stable master cell banks capable of virtually unlimited expansion and differentiation into any cell type. Our scientists can program the iPSCs to carry out specific functions. For example, delivering lost signals in the brain, pumping blood in the heart, or activating a therapeutic immune response. Additionally, we can further engineer the cells to enhance their capabilities by gene editing. For example, we can avoid recognition and thus rejection by the patient's immune system. Gene editing can further enable the cells to deliver a payload of choice, improving the therapeutic effect. This opens the door to creating medicines that raise the standard of care and enable therapies for previously intractable diseases. With our iPSC technology, we are aiming to redefine the very way we treat diseases, moving from addressing mere symptoms to restoring health lost to cell damage or degeneration. by engineering the cells to work seamlessly with the surrounding healthy cells to restore function. Summarized, iPSC technology allows us to create a virtually unlimited supply of almost any cell type that can then be used to restore health in patients suffering from severe diseases. This opens great potential for developing novel therapies with long-term clinical benefit. To date, there were no approved iPSC-based products on the market. Clinical activity is still in early stages, and we expect the first phase 3 trials to start in the next few years.